Okay, true or false review for grade 11 biology exam. This is going to utilize the true false handout that you guys should have if you're in my class. And if you don't, then you could just follow along with this and see how your grade 11 biology course compares to the one that I've been teaching. The first thing that I want to say about this is some of the questions were submitted by other teachers in the department, and so you might see some stuff that's not exactly stuff that we covered in class. So don't worry about that too much. Just get what you can out of this and follow along. And here we go. Okay, so you should have your true or false question sheet. And I'm sorry if this looks a little blurry to you. I hope that you can read this. You should, of course, have the sheet there. It might be a little blurry on the screen. I'm not going to be able to go through this in any great extent. This is just a review, so I'll try to scribble here and there and see how it goes. Okay, so I am going to do some writing here, or maybe over here if this works. Let's see if I can switch between them. This kind of thing. There. Do that kind of thing. I'll scribble away here. PowerPoint. Oh boy, hang on a second. How do I get out of that? There we go. Okay, and so here we go. So question number 1 in the genetics section is that skin cells are examples of somatic cells. And that's true, of course. You've got somatic cells and you've got gametes. The somatic cells are the body cells. So this one is true. Metaphase is the third stage of mitosis. So, well, we've got, of course, interphase. That's not part of mitosis. That's just part of the cell cycle. And then as far as mitosis goes, we've got prophase, metaphase, anaphase, and telophase. So metaphase is the second stage, so that is not correct. Interphase is the longest stage of mitosis. Well, it's the longest stage of the cell cycle. So if you remember the cell cycle, we got this kind of thing, right? And most of the time, the cells are in interphase, and then sort of around there, to there, they're going to be in mitosis. So all of this stuff is going to be interphase going around there. So interphase is the longest stage of the cell cycle, but it's not the longest stage of mitosis, so that's false. DNA replication occurs during interphase. That is true. DNA replication occurs sometime here or here, replicating the chromosomes for mitosis. So that is going to be true. It is part of interphase, and then the cell is going to go into mitosis itself. Next one, chromatids are held together by centrioles. All right, so I'm going to clear all this, so make sure that you've got this. So periodically I'll be drawing some stuff, and then of course I'm going to lose all the text here. So I'm going to clear this. I'm going on to number 5. Okay, chromatids are held together by centrioles. So we've got the cell over here. And I'm just going to focus on the nucleus right now. The chromosomes start out like that. They're long stringy things, chromatin, and then during interphase, when they replicate, you're going to get this sort of X type structure there. Each one of these is a replicated chromosome, but they're called chromatids. Joined together, they're going to be called sister chromatids, and they are going to be held together by a structure there in the middle, which is called a centromere. The centrioles, on the other hand, are structures that are going to be, usually at the poles, whatever, and they're going to create the spindle fibers, which are going to attach to the chromosomes, or to the chromatid, to the centromere, really, holding the chromatids together. So the chromatids are not held together by centrioles. They're held together by centromeres. So number 5 is going to be false. Number 6, during anaphase, chromosomes line up along the equatorial plate. All right, and this can really apply to either mitosis or meiosis. I'm getting rid of that. This is also not true because the chromosomes line up during metaphase, and that's where we're going to see all the chromosomes lined up, roughly in the middle of the cell, that kind of thing. So this is metaphase when they're lined up, and of course during anaphase, that's when the chromosomes are going to be pulled apart. Either the entire X structure, which is the homologous chromosome, or they're going to be pulled apart. The chromatids will be pulled apart in anaphase of meiosis. That would be anaphase 2 when they actually get pulled apart, anaphase 1 when the X moves. But this is not true. Number 7, cleavage furrows occur in plant cells and cell plates form in animal cells. Well, this may not be something that I discussed a lot in our class, but it's not hard to do. In telophase, you've got, the recreation of the nuclear membrane, and the chromosomes are all in here. And then at some point, the cytoplasm is going to pinch in. Now, we refer to that as cytokinesis. And in an animal cell, when that happens, they sort of start pulling apart like this, right? So they got a nucleus there, you got a nucleus there, and that is called furrowing as it dips down, and that's called a cleavage furrow. This will eventually pinch apart, and you'll have two separate cells, and they'll either go on their own way if they're sort of motile or mobile cells, or you may have cells that will separate and somewhat stay together and forming a tissue. All right, but it's still formed by cleavage, or that cytokinesis. In plant cells, however, you recall if you've seen the onion root mitosis, you get these rectangular cells in a brickwork type fashion like that. When the cells separate, they form what's called a cell plate, sort of a cell wall separating the two cells. So this is actually backwards. The cleavage furrow is in animal cells, and the cell plate is in plant cells. So number 7 is false. Spindles project from centrioles. I would prefer to use the name spindle fibers, but this is generally true, right? If the centriole was up here, the spindle fibers come sticking out like that, and they attach to the chromosomes like that. So we could say that this is essentially correct. Nuclear membrane reforms during telophase. Yes, that's true. We already talked about that. During telophase, the nuclear membrane is reforming around the chromosomes, and then eventually we'll have the cytokinesis. Okay, number 10, cytokinesis refers to division of cytoplasm at the end of mitosis. Yes, that's basically true. Cytokinesis, the separating of the cells. Okay, a little bit of genetic biochemistry now. Nucleotides are the basic building block or monomer of DNA. Yes, that's true. So, of course, DNA is this double helix structure like this, and along the double helix we have the nucleotides. The nucleotide is made up of a phosphate, a sugar, and a nitrogenous base, and this is the building block of DNA, the nucleotides. So that's correct. Number 12, the DNA backbone is comprised of sugars and nitrogenous bases. All right, well, I don't have a really good picture of this, but I mean, if you were looking at the ladder-like structure of DNA, you've got something like this. All right, I think my dogs are going to come in here. You got this kind of structure, and projecting into the middle here, these are the bases. So, for example, if you had adenine here, this one would be thymine. If you had cytosine here, this one would be guanine. Each of these nitrogenous bases are connected at this point here, okay, right there. That's going to be the sugar group, all right? And that sugar, of course, is deoxyribose. So it's a deoxyribose sugar, and let's see, let's switch to this color, and right there, that doesn't exactly look that clear. You're going to get another part, and that's going to be the phosphate group. All right, so between the three of those, the base, the sugar, and the phosphate group, well, there's your nucleotide. All right, so the DNA backbone itself is this part here. All right, not including the base, and that's a sugar phosphate backbone. So let me switch back to my red. So the DNA backbone is comprised of sugars, yes, but not nitrogenous bases, phosphate groups. So that's wrong. Cytosine binds with guanine. Yes, that's true. C and G and A and T. So there's our cytosine. It bonds with guanine, whereas adenine bonds with thymine. Number 14, pyrimidines always bind with other pyrimidines. All right. So in the DNA nucleotides, as you know, there are 4 different kinds, right? You've got adenine, thymine, cytosine, and guanine. We're only talking about DNA. We're not talking about RNA nucleotides. So we're not involving another base, which is called uracil. Some of these are called pyrimidines. So this one is called a pyrimidine, thymine, and cytosine is called a pyrimidine. And a good way to remember that is, of course, thymine has a Y in it, cytosine has a Y in it, and so does the word pyrimidine. These other ones are called purines. So that's a purine, and this one is a purine. All right, and if you look at their actual molecular structure, you can see ring structures like this, and when you have two rings together, this is a purine, and when you have a single ring as part of the base, that's a pyrimidine. And so it's kind of weird because the smaller ring is the longer word, pyrimidine. The double ring is the smaller word, so it's the inverse. And so what happens is pyrimidines bind with purines, not with other pyrimidines. So if this is a pyrimidine, again, it's just a family of molecules like carbohydrates, right? But then there's specific carbohydrates. Well, there are specific pyrimidines, and thymine is one of them. Thymine, of course, binds with adenine. Cytosine, of course, binds with guanine. So it's a purine and a pyrimidine. So this is false. Proper base pairing is referred to as complementary base pairing. And yes, that is correct. Adenine, thymine, cytosine, guanine, they fit together. They're held together by hydrogen bonds, and this is called complementary base pairing. Number 16, nitrogenous bases are held together by hydrogen bonds. Yes, that's true. So if we really do, I'm going to just make sure you know that's true, number 16. I'm going to get rid of that. If we have adenine and thymine and cytosine and guanine, hydrogen bonds are illustrated using dots or dashed lines, and there are two hydrogen bonds between adenine and thymine, whereas there are three hydrogen bonds between cytosine and guanine. Good way to remember that, three hydrogen bonds, C is the third letter of the alphabet. Or two hydrogen bonds, well, A is very early in the alphabet, so it's the lower number of hydrogen bonds for the lower letter in the alphabet. So this is true. They are held together by hydrogen bonds, and this, of course, allows them to be separated relatively easy by the helicase enzyme in DNA replication. Okay, number 17. A polynucleotide is represented by half of the DNA molecule. Well, yeah, so here's the thing. Okay, DNA, right? You got sugar. There's a sugar connected to our base, and there's our phosphate group. All right, and phosphate group, sugar, sorry, and another base. Phosphate group, sugar, and another base. All right, as you know, the nucleotide is the building block of DNA, and DNA is a strand of nucleotides. You can have, of course, two sides to this thing. And really, I mean, whenever you have a long connection of things, whether it's starch, and we're looking at glucose connected together like this, or glycogen, or even amino acids connected together, you get a polymer, P-O-L-Y, poly referring to many. So, where am I? A polynucleotide basically just means you've got a bunch of nucleotides. So a bunch of nucleotides does not have to be half of the DNA molecule. It could be the whole thing. I mean, this right here is essentially a polynucleotide, more than one nucleotide, or a bunch of nucleotides. So half of the DNA is a polynucleotide. The whole thing is a polynucleotide. 3/4 of it is a polynucleotide. Kind of a dumb question. Wrong. Okay. Gyrase. Now, notice how this is written. I mean, I was talking about this in some of my classes, maybe even to you guys. We're mentioning the different enzymes in DNA replication. And it says here, gyrase and helicase is responsible for unzipping and unwinding the DNA molecule during replication. Yes, that's fine, all right? Now, I specifically focused on helicase, which splits the hydrogen bonds between adenine and thymine, cytosine and guanine, all right? I'm not drawing the rest of the DNA, right? That's helicase cuts there, okay? Well, there's the strands there, that kind of thing, all right? Now, the thing is that DNA is actually a coiled molecule, double helix. I shouldn't have done it that way. Whoa, that's not good, right? You guys are laughing at my artistic ability, I'm sure. Oh, whatever. There we go. That's a little bit better. Okay, so, you know, and you've got the hydrogen bonds in here with the bases. And so, sure, it's one thing to go through and to cut the hydrogen bonds there, But notice that this thing is coiled up. So imagine if you cut here and you sort of pulled this piece apart and pulled that apart, right? And you're doing that. As you pulled it apart, it actually gets tighter and tighter because it's wound up. So there is another enzyme, and it's called gyrase, and it actually untwists, the DNA molecule. It's twisted like that. It untwists it, sort of separates it out so that you can cut up the middle. It's not critical for grade 11. Helicase is the one I'd rather focus on as the enzyme that really unzips. That's the cutting, of the hydrogen bonds, unzipping the DNA molecule. But unwinding is done by gyrase, and together they do that first step. So I would prefer that you know about helicase, but gyrase can be another enzyme that is involved with DNA replication. So that's true. Okay, number 19, DNA polymerase attaches the free nucleotides to the growing DNA molecule. And yes, that's true. So when you do unzip this and you have one strand here with all of its bases sitting there freely without the partner, right? You now need the complementary base on there. You need the complementary base on each of these. And the enzyme DNA polymerase comes along and finds a cytosine in the nucleus, clicks it onto a guanine, finds a guanine, clicks it on an adenine here, a thymine here. So DNA polymerase is the enzyme in the nucleus that is doing that second step. We'll call it the second step anyhow. Helicase for the first step, DNA polymerase for the second step, and then of course the last step is going to involve ligase, where once these bases are clicked on, these are all individual nucleotides. We have to create this sugar phosphate backbone. We have to click them together in the row, and that involves ligase, and that's another enzyme. So that's true. Number 20, chromosomes that occur as a pair are termed homologous chromosomes. Okay, that is true. And so you've got chromosome 1 from father, chromosome 1 from mother, chromosome 2 from father, chromosome 2 from mother. And of course, these all have the alleles on them, the genes, the similar genes. So let's say you've got ability to roll the tongue or not roll your tongue. Whatever, doesn't really matter. Tallness, shortness. And I mean, you can have the same gene as well. So for example, the gene that produces insulin in dad is the exact same gene that produces insulin in mom. And so if this is on chromosome number 1, let's say, and I don't know what chromosome it's on, this will be the exact same gene. But you can also have variations of genes, which are called alleles. So, dad has brown hair, mom has blonde hair. Dad has green eyes, mom has blue eyes, that kind of thing. Okay, but the chromosomes in a pair are called homologous chromosomes because they have similar genes. Whereas chromosome 1 and chromosome 2 are not considered homologous chromosomes. They don't have similar genes. Number 21, the most active stage of meiosis is metaphase 1. Well, let's say no. Now, obviously, this brings in some discussion. So we're saying here, where am I? The most active stage. We're saying false. Think about what you know about metaphase 1. Okay, what's going on in metaphase 1? Well, the chromosomes, of course, are replicated. This is meiosis. So they're like this. And they are paired up with their homologous chromosome. This is a homologous pair. This is not two homologous pairs, all right? This is one homologous chromosome, one homologous chromosome. However, each one of these is doubled, as you can see, but it's still called just a homologous chromosome. So this is one homologous pair of chromosomes. This is another homologous pair and another homologous pair, all right? During metaphase 1, the homologous pairs are lined up in the middle, but they are undergoing crossing over. So the non-sister chromatids here are crossing over. These non-sister chromatids and this part. And it can even happen between this one and the outer one there, if that's the outer one, okay? These are called non-sister chromatids, and they undergo crossing over. Now, this is an active process of synapsis, crossing over at a chiasma, exchanging genetic material. But I mean, I don't think you would call this the most active phase. You could argue that. But then after that, of course, the chromosomes are going to be pulled apart during anaphase 1. The entire X is going to move, all right? And this entire X is going to move, and that's really anaphase 1, and that's probably considered more active. You know, you could also argue prophase 1 is a pretty active stage, because during prophase 1, right, you've got the cell here, the nuclear membrane is breaking down, spindle fibers are being produced by the centrioles, and crossing over is occurring once the homologous chromosomes find each other. So they're finding each other in prophase 1 and crossing over. You could argue that prophase 1 is definitely more active than metaphase 1. Okay, so that's the idea behind that one. All right, let's go on here. Where are we? 23. The area of crisscross. No, I'm not there. I'm on 22. Okay, so this one we're saying false. 21. 22. During prophase 1, non-sister chromatids cross over and exchange genetic material. Prophase 1, non-sister chromatids. Yes, that sounds good. We just talked about that. 23. The area of crisscross during meiosis is termed chiasma. I'm not crazy about the grammar of that one, but it's basically true. So you've got a homologous chromosome there. You've got its partner over here. All right. I'm going to get crazy with some colors here. And let's go with the green. And these two are going to cross over here. I should draw these a little bit different. Let's actually draw them going like that. Okay. And at that spot there, that is called a chiasma. That's the point at which the non-sister chromatids overlap or touch, and that is where crossing over is going to occur. That's the area of crisscross. All right, so that's good, chiasma. And of course, they can cross over at multiple spots, many areas, so many chiasmata. Number 24, meiosis reduces the chromosome number. Yes, that's one of the key things about meiosis in humans. If we start with 46 chromosomes, we will eventually end up with 23 chromosomes, the haploid number in the gametes, whereas 46 is the diploid number. Okay, hang on. My dog is crying here. Let's pause this. Okay, sorry about that. My dogs were looking for some attention. So where were we? Okay, so anyways, yeah, meiosis reduces the chromosome number, converts diploid cells to haploid gametes. Okay? Number 25, meiosis takes a diploid cell and ends with 2 haploid cells. Well, that's not correct. First of all, if we're talking about the production of sperm, we're going to get 4 haploid cells. All right? So that's wrong for that reason. And so it's going to be wrong. And if we're talking about the production of eggs, we're only going to get 1 haploid final egg and 3 sort of smaller polar bodies that contribute their cytoplasm to that, and their DNA is just recycled. So that's not true. Meiosis 2 is very similar to mitosis. That is true. The second half of meiosis, if you follow along with your old diagram, it is very similar to mitosis, so that's true. Spermatogenesis involves the equal division of cytoplasm. That's essentially true. That's just getting at the fact that you produce those 4 haploid gametes, equal in size, obviously equal in chromosome number. Whereas oogenesis here, the production of the eggs, is of course going to be different. Okay. Number 28, cloning creates identical cells. Therefore, mitosis is an example of natural cloning. Cloning creates identical cells. Mitosis is an example of natural cloning. Yeah, essentially that's true. Obviously cloning, it's a long process, but when you start with one organism and you clone it, and you end up getting an identical organism, okay, or an identical cell. All right, because you're taking the nucleus from the somatic cell. It all starts with one cell. You take that nucleus, you put it in the enucleated unfertilized egg. So you put the nucleus in there, and let's say you make another one. Okay, it divides into another cell. You're getting an identical cell here, and so the same thing is happening in mitosis. So that's why that's true. All right, to clone an organism, one requires an enucleated egg and the DNA from the cell to be cloned. Clone an organism, one requires an enucleated egg. Yes, that looks good. And the DNA from the cell to be cloned. Yes, the donor cell. Okay, so that's 29 is going to be true. All right. Number 30, the cell cycle of a cancer cell is much faster than a healthy cell. This is something that you usually talk about in grade 10. Usually talk about cancer and the fact that it's mitosis out of control. So, you know, the thing that I'm not sure about, to be quite honest with you, is we know that in cancer, so there's something genetic in that the cells just continue to divide and divide and divide, and they don't stop undergoing mitosis as they normally would. All right, and so you end up getting this mass of cells, which is a tumor, which crowds out cells, robs cells of nutrients, and that kind of thing. And so it would seem that the cell cycle is faster in these cells, or just the fact that yeah, well they don't stop dividing. So I'm going to go with true. Okay. Number 31, some chromosomal disorders are the result of non-disjunction. Chromosome pairs do not separate during meiosis. Yeah, now I want to delete this stuff. And so we're on 31. And so I'm going to go for true. Some chromosomal disorders are a result of non-disjunction. Okay, and this is when, you know, chromosomes don't separate properly. So you got them here like this. And this one's supposed to go that way. And this one's supposed to go that way. And this one goes that way, but this one also goes that way, and you end up getting like a, you know, a trisomy or something like that. And so that's a non-disjunction, and the chromosomes don't separate properly during meiosis, and therefore the gametes that are produced have the wrong number of chromosomes, and so when the gametes fuses with the other gamete, you have that genetic disorder. The human haploid number is 46. That's not true. The human haploid number is 23, and the diploid number is 46. Okay, so I'm going to move on to my next page now. And we're still in genetics, but now we're getting into sort of Mendelian genetics. All right, here we go. The dominant trait is normally expressed in the heterozygous form. A heterozygote is something like big T, little t, as we can see here. That is heterozygous. The big T is dominant, the little t is recessive. But the dominant trait, which is coded for by the big T, let's say tallness, can be expressed in the heterozygote. Yes, it can be, but it can also be expressed in the homozygote or homozygous condition of big T, big T. So it's not normally expressed in the heterozygous form. It can be expressed in the heterozygous form, and it can be expressed in the homozygous form. So this is not correct. A homozygous condition exists when two alleles are different. No, that's not true. This is heterozygous right here. This is homozygous. So homozygous is big T, not big T, little t. False. A blending of traits occurs during incomplete dominant crosses. And that's right. When an allele or a gene is not completely dominant, as in this case where the big T masks the little t and you've got just tall. If you had a situation where phenotypes could be tall or short, and of course in order to make short you'd normally need little t. But if you've got this intermediate thing and you end up getting sort of medium height, then that would be called incomplete dominance. And this is essentially a blending of the traits. And we really see this. I'm going to say, first of all, that this is correct. Okay, so mark that one as correct. We normally see this with the flowers, with like the snapdragons, where you have the red color and you've got the white color, and an intermediate color is pink because those ones blend together and you get the pink. And so in the Punnett square of something like that, you've got big R's there, and you got the big W's there, and you know whether or not we're using other letters to represent this. In this case, you're going to get this, right? And that kind of thing is an intermediate between red and white, and this would be represented as pink. So, if you were looking at a Punnett square and you had this, sorry about that, big RW, what's that going to give me? Big R, big R, big RW, there we go, big RW. WW. So when you end up getting a different kind of ratio than you would have expected, like for example, normally we would say, okay, we have 3 of this type, and there's our homozygous recessive there, usually, all right? You get 2 different phenotypes. But in this case, we get red, we get white, or pink, sorry, and I'll use a diamond shape there, whatever, and we get the white. So red, pink, and white. We get 3 phenotypes here instead of 2, all right? That's a hint that this is an incomplete dominance type situation. So that was 3, that was true. Number 4, crossing an organism with a homozygous recessive organism defines a test cross. Yes, now this is the test cross scenario, excuse me, that we sometimes struggled with in class. If you've got an organism and it's tall and you don't know if it's big T or little t, and so we use a dash. If you need to know what that other gene is, you can do a test cross. You cross it with a homozygous recessive, and you get your Punnett square like this, big T dash, little t, and you get big T, little t, big T, little t. I don't know how clear that is. Dash, little t, and then you look at the offspring, and if all of the offspring are tall, this has to be a big T. And so 100% tall, we now know that that's a big T. But if these ones are short, right, if those ones are tall and these ones are short, we know this has to be a little t, in which case that's a little t. So this is called a test cross. You take your unknown organism, which with an unknown genotype, you cross it with a known genotype. This has to be homozygous recessive, and then you look at the offspring. If half of them are show the recessive trait, then you know that's a little t. Okay, so number 4. We are going to go with test cross. True. Number 5, any characteristic has no more than 2 traits. This is not true. And it's not true because of the reason I just showed you with the incomplete dominance, right? When we had this, right, we got potentially 3 any characteristic. 2 traits. Well, we got 3 phenotypes, or we could say 3 traits. Wording of this is awkward. It's not the same wording as I usually use. That's 1 trait. That's the red trait. This is the white trait, and this is the pink trait. Well, that's 3 traits. Whereas normally, in complete dominance, if we did the same kind of cross, but using complete dominance and heterozygotes, we get this, right? And this is what we started out with when genetics was fun, right? Fun and easy. In this case, you'd say, okay, there are 2 traits. There's the tall, and there's the short, and that's all there is. But when you start dealing with this, you get 3 traits. And what about blood type? There's type A, there's type B, that's 2 right there. There's type AB, well, there's 3. There's type O, and there's even positives and negatives, which I didn't get into this year. So this is not true. Any carry more than 2 traits? No, there's more. Genetic crosses involving 2 characteristics is termed a dihybrid cross. Yeah, and that's basically true. Like, you've got an organism that's tall, and let's say it's heterozygous tall, and it's homozygous dominant round seeds, let's say. That's 2 traits, tallness trait, roundness trait. So 2 characteristics or 2 traits, that is dihybrid, whereas these are monohybrid. Genes located on the X chromosome are termed sex-linked. Yes, so you've got your 22 pairs of autosomes, and you got your final pair of chromosomes. Those are the sex chromosomes, right? You got an X chromosome, and if it's XX, it's female. If it's XY, it's male. And chromosomes or genes or traits that are on the X chromosome are called sex-linked. There are sex-linked genes on the Y chromosome, but we haven't talked about them. There's few of them. Typically, we talk about X chromosome genes, and they're called sex-linked. So that is true. Okay, I'm going to get rid of this to clean it up. Number 8, a karyotype is a picture of all the chromosome pairs of an individual cell. Yeah, that's true. So Remember your karyotypes, you got this, you got 4 rows, sex chromosomes are down here, let's say, all right, and they're organized into pairs, all right? So there's a pair, and here's another pair, whether they're doubled or not doubled. So that's true. Watson and Crick are the fathers of genetics. Well, you know, that's not really true. They are the ones that discovered the double helix DNA structure, or got the Nobel Prize for finally figuring it out. And usually Mendel is considered the father of genetics. 10, genotype refers to the physical description of a gene. No, the phenotype refers to the physical description of a gene, or what the gene codes for and what it causes, what its expression is. Genotype is the actual letters of a gene. Once again, I find this awkward phrasing. If you do as well, then I will not be surprised. But this is just not the way I explained it in class. If another teacher may have explained it that way, then it makes more sense, but I don't really like it. On the other hand, you know, if this is meant to imply physical description of a gene, if this is meant to imply, okay, this gene is a big T gene and this one is a little t gene, that's the physical description, whereas, you know, the expression of the gene, causes tallness and this causes shortness, then, you know, you could look at it another way and say, well, it's right. You know, this is not the kind of problem that you should run into on the exam, all right? You're going to do it from our perspective, from what I taught you in class, and the questions should make sense, and they're not going to be ambiguous. Or if it is an ambiguous type thing, it's something that you're supposed to discuss and explain. All right. A 3-to-1 offspring phenotypic ratio occurs in a monohybrid cross between 2 heterozygous parents. So that's generally 2. And that's what we've been looking at. Big T, little t, big T, little t, big T, big T, big T, little t, big T, little t, little t, little t. 3-to-1 ratio. Check. Okay, now look, number 12 is true, all right? These are some typical ratios that you will see. A 9-to-3 to 3-to-1 offspring ratio, phenotypic ratio in a dihybrid cross between 2 heterozygous. In a dihybrid cross like this, if you had this, go ahead and do this sometime, or go back in your notes and you will see those are 2 heterozygotes. They are completely heterozygous in a dihybrid cross. If you do this, 16 offspring, you will get a 9-to-3 to 3-to-1 ratio in terms of phenotype. Like, you know, 9 tall and round, 3 tall and wrinkled, 3 short and round, and then 1 short and wrinkled at the very bottom of the Punnett square. So this is also true. And we've all already seen this ratio as well a number of times with the red and the white flowers. If I cross them, I got up here, I got 1 of those, and then I got 2 of those, and then I got 1 of those bottom ones. So that is also true. So all 3 of these are true, and they're pretty typical standard genetic ratios. So let's go on. Number 14, a trait that is governed by more than 1 gene is referred to as a polygenic trait. I didn't cover this in class, but there are traits that are caused by more than 1 gene. We looked at things like eye color and we said, okay, if you have big B, you have dark eyes. If you have big B, you still have dark eyes. And if you have little b, you have light eyes like blue eyes. There are more genes actually that determine eye color than just this, but this is the way we do it in grade 11. This is called polygenic, and this is true. This is not something that you typically have to worry a lot about though. Okay. Number 15, human blood groups are an example of codominance. That's true. And the reason is because we can have the AB blood type, right? Where you've got a blood cell that's got some A's on it and some B's on it. This is a red blood cell, and this is called codominance, where A and B are equally dominant. Now, of course, when you do this, you use this kind of nomenclature or this kind of writing for that, okay? the IA and IB, and of course the little I if you wanted type A blood, but with the recessive gene, you draw a little I. Okay, hang on, time out for a second here. All right, number 16. And female with genotype X big C, X little c for colorblindness is colorblind. All right, well, now we're talking about a sex-linked disorder, colorblindness. We are looking at a female genotype, big or XX, that's female. Colorblindness is generally a recessive sex-linked trait. And here you can see the little c, which would cause colorblindness. But of course, this female has the dominant gene to mask the recessive gene. So she is a carrier for colorblindness. She's heterozygous. She carries the gene for colorblindness, but she is not actually colorblind. So number 16 is going to be false. In order for her to be colorblind, she would have to be X little c, X little c. Those are littles as opposed to X, say big C, X little c, right? This is the condition for her to be colorblind. Here she is just considered a carrier. And so in a pedigree, her circle would be drawn half filled in like this, and then you would write X big C, X little c. Now, if you didn't know if she carried the gene, she would be X big C, X dash, all right? And we wouldn't know what to do for her pedigree symbol exactly. A male, on the other hand, of course, if he has just one little c, because he doesn't have the gene on the Y chromosome to mask it, this male would be colorblind and would be completely filled in on a pedigree. Number 17. A person with blood type O has type A and type B antigens on their blood cell membrane. Well, that's not going to be true, because if this is the red blood cell, when a person has type O blood, it means they don't have any of those A or B antigens or glycoproteins on the red blood cell. All right. If you have type A glycoproteins, then you have type A blood. And if you have B glycoproteins, you have type B blood. And if you have both, you have type AB. These things are referred to as antigens because they can trigger an antibody response. But if you have type O blood, you don't have this, and you don't have this, so you don't have these antigens. You don't have these glycoproteins around to trigger an antibody response. So this is not true. A person identified as XXY is female? No, because when there is the X chromosome and a Y chromosome, they are referred to as male. So this is not true. And that would be the Klinefelter's syndrome right there, right? Number 19, a person identified as X0 or XO has Turner's. Yes, that is true. This is a monosomy, sorry, where there's only one sex chromosome. The other sex chromosome didn't come into that cell, and so this is Turner syndrome. So they are female, and they have their various symptoms that you should be familiar with. So that's true. Number 20, a person with trisomy 21 has Down syndrome as a result of non-disjunction and meiosis. That's true as well. This is trisomy 21, so they have 3 chromosome number 21s instead of just 2, and that extra chromosome would have come over in the sperm cell or the egg cell, and that causes Down syndrome with all of its genetic characteristics. Pedigrees are family trees that show genotypes of related individuals. Yes, they do. All right, so if you've got a father here, and you've got a mother, and then they have some kids, there's a daughter, and a son, sorry, over there, and so then you just look at, father's got these genes, mother is these, and then they've got the daughters, and you got the son, and whatever genes are on there, and whether they're half-filled or full-filled or whatever, but they do show the genetics of related individuals, and we've done a bunch of those, and I've got some video tutorials on that as well. Okay, I got to think about this one now. A person who is blood type AB positive can receive blood from a person who's O negative. Now, I didn't get into the positive and the negative, and I wouldn't worry about that. It's more a question for now, can a person with AB, so let me just write this out, person with AB blood receive from a person with type O? So what we're saying is, can this blood go into this blood? All right. Now, the positive and the negative do have implications on this question. I just want to omit them and see what we can do if it's like this. If the person has AB, that means they have A and B glycoproteins or antigens on their red blood cells. That means they're not going to create anti-A and they're not going to create anti-B, or else they attack their own cells. If blood type O comes in, well, they don't even have the antigens or the glycoproteins anyhow. So even if these antibodies were present, there would be nothing to attack. And so this blood could be donated, not including the positive or the negative. I'm going to put a question mark for that one. Number 23, females who are carriers for sex-linked traits always express the trait. Well, that's obviously not true. You've got a female here who carries the recessive gene for hemophilia. She doesn't suffer from it. All right, so that's the whole concept of a carrier. So this is not true. Humans have 22 pairs of autosomes. That is true. 23 pairs of chromosomes. The first 22 are autosomes. The last pair sex chromosomes. That is true. Amniocentesis can test for chromosomal abnormalities. Yes, and that's where they go in with the syringe and they remove the cells from the baby, and they're going to, potentially create a karyotype. And so amniocentesis is the use of the syringe, I guess, and maybe the ultrasound for viewing the baby to get those cells so that you can test for chromosomal abnormalities. So that's correct. And we did not cover chorionic villus sampling, so we're not going to worry about it. It's another testing technique on the baby, or the unborn child. All right. Okay. We're going to go on a little further, but it's going pretty long, isn't it? I'm just going to do a little bit more of this for you, and the answers are already available, so you can always come talk to me some more if you want to as well. So I'll just sort of speed up a bit. Protein, carbohydrates, and minerals are all macronutrients. This is not true, because minerals are micronutrients. They are needed in small amounts every day, like vitamins. Minerals include things like iron and calcium and magnesium and zinc and stuff like that you would see on your vitamin pill. All right. And so these minerals are micronutrients, not macronutrients, whereas proteins and carbohydrates are macronutrients, as well as lipids. Proteins are made of amino acids. Yes, that is true. Numbering is all weird here. Number 5, we're jumping around. Most energy comes from carbohydrates. Yes, that's true. Glucose is the major, we could argue this a little bit. Glucose is the primary energy source in the blood. The cells use cellular respiration to break down glucose. Now, other carbohydrates can be used as well. You could use fructose. You'll see this in grade 12. You could use galactose. You can use many different monosaccharides. You can break your disaccharides into monosaccharides and use them in cell respiration. You can break your polysaccharides, starch, or glycogen down into monosaccharides and use them for energy. It's usually carbohydrates. However, fats actually store more energy and can provide more energy if they are burned, used, metabolized in proper situations. This is more grade 12 concept, and you'll see that, and you should understand that in grade 12. However, I really think that for grade 11 biology, we're getting at the concept that most energy comes from carbohydrates. Glucose is our primary energy source. So we're going to go with true for that. Number 6, enzymes are involved in physical digestion of food. That's not true. They are involved with chemical digestion of food. Number 7, enzymes lower activation energy needed to start a chemical reaction. This will be a grade 12 concept as well, or you may have seen some of this in grade 11 chemistry. In order to make any chemical reaction go, energy has to be put in. So to get these molecules to react, you have to put in some energy to get them to react. Enzymes actually help make molecules react, as you know, right? Or break stuff down, right? They can break the bonds and break stuff. It takes energy to do that, and that energy is called activation energy. All right, it's not a concept that we really talked about, but it is true. An enzyme binds to a substrate and forms an enzyme product complex. All right, so in the breakdown, let's say, of starch, right? You know that's going to take the enzyme amylase. And in order for amylase to break down the starch, they have to come together. So amylase is going to be a big molecule here, a big enzyme. That's the enzyme amylase, and it's going to break down the starch. When they come together, this is called an enzyme substrate complex because starch is the substrate for the enzyme amylase. So this is called an enzyme substrate complex. Amylase will do its job, break the starch into maltose. The maltose is the product. So to say this, an enzyme binds to a substrate and forms an enzyme product complex, that's kind of ridiculous because the enzyme will bind to the substrate and then release the products. In this case, it could be maltose molecules. They're gone. They're released. They're not attached to the enzyme. So this is false. Okay, let me clean this up. Number 9. The substrate for protease is protein, and the products are carbohydrates. Now, you need to be able to understand this enzyme stuff and think it through. This is not overly complicated, all right? Protease is a general name for enzymes that break down proteins. Pepsin is a protease because it breaks down proteins into smaller building blocks, which essentially would be polypeptides or peptides. If you took erepsin, let's say, it breaks down polypeptides and it breaks them down into individual amino acids. These are proteases. They work on proteins, all right? The products are not carbohydrates, though. The products are either individual amino acids or they're polypeptides or peptides, okay? So that's why this is wrong. The substrate is protein. All right, so for example, the substrate for pepsin is protein. Substrate is the thing that's acted on. The products are not carbohydrates, though. So number 9 is false. Number 10, enzymes can be harmed. Number 10, enzymes can be harmed by changes in temperature and pH. Yes, that's denaturation. Pepsin is an example of a lipase. No, that's not true. Pepsin is a protease. It breaks down proteins. Lipase breaks down lipids. Bile is produced in the liver and stored in the gallbladder and breaks down fat. Yes, we like that one. Humans do not possess canine teeth. That's not true. Humans do have canine teeth, although we are essentially omnivores. We're carnivorous and herbivorous at the same time. The sublingual gland is a salivary gland located right beneath the ear. Well, although we didn't concentrate a lot on the locations of them, you probably know that it's the parotid gland that is behind the ear, the big one. And of course, sublingual means under the tongue, sublingual. So that's not true. Amylase acts on starch and produces maltose. Good. The esophagus has 3 muscle layers. No. Okay, the esophagus is going to have its lumen there. It's going to have a mucosa, a submucosa, and then in the muscularis, it's going to have 2 layers, circular and longitudinal. The stomach has the 3 layers. The submucosa is the layer most closest to the lumen in the esophagus. No, that's not true. If that's the lumen, we have mucosa, then submucosa. So that's not true. Number 18, the pyloric sphincter controls entry of food to the stomach from the esophagus. That's not true as well. When food enters the stomach here, right, it goes through the cardiac or the esophageal sphincter. The pyloric sphincter is down there. The stomach possesses villi. No, villi are in the small intestine. The stomach has rugae. Gastric juice is created in the gastric glands in the stomach wall. That's good enough. All right, stomach wall is a little bit vague, but those are the gastric glands are in there, and they, of course, release their contents into the lumen. All of these are in gastric juice. Let's check it out. HCl, yes. Pepsin, yes. Bile, no. That's from the gallbladder or the liver. Lipase, yes, although there is also lipase from the pancreas, so this becomes false. The 3 sections of the small intestine in correct order are duodenum, ileum, no. Jejunum, no. These are reversed. Glucose and other monosaccharides enter the lacteal of the villus. No, that's not true as well. So when you've got your villus here, you've got your lacteal in the middle, and you've got your capillaries that are surrounding it. Glucose and amino acids enter the capillary network, and fats, like glycerol, fatty acids, that kind of thing, enter the lacteal. So it's false for that reason. Blood enters a villus through a venule. No, the blood vessel bringing blood in is called the arteriole. The blood exiting the villus once the nutrients are absorbed is the venule. So this is wrong. Your ascending colon is on the right side of your body. Okay, well, the ascending colon goes up here. we go across transverse, and then we come down. So this is the left side here. This is the right side of my diagram. So they would be reversed in the human body. And so, yeah, that's true. Okay, I want to clean this up. Just going to go for another minute or two. The colon produces vitamins and absorbs most nutrients. It does produce some vitamins, and of course it absorbs water. That's the main thing. It doesn't absorb most nutrients. That's done by the small intestine. So that's going to be false. Inability to absorb water from the GI or gastrointestinal tract could imply a problem with the large intestine. Yeah, and that's true. That's what I was just saying. Okay. Overall chemical digestion begins in the stomach. No, it's not true. It begins in the oral cavity. So let's not even go any further with that one. 29, the small intestine possesses the same basic layers as esophagus. Yes, that's true. I mean, same basic layers meaning mucosa, submucosa, muscularis. But there are, of course, differences. The villi are present, but that's not what the question is asking. Insulin is an enzyme. Stop. It's not true anyhow. Insulin is a hormone. It is produced by the pancreas, and it does act on the liver, right? And insulin, of course, helps the liver absorb glucose and store it as glycogen, but it's not an enzyme. It's a hormone. Glucagon is a hormone made by the pancreas, raises glucose by causing the liver to release glycogen into the bloodstream. Well, we got to be careful. Glucagon is a hormone. It's produced in the pancreas. It raises glucose level, but it's really not by this mechanism. It causes the liver to take the glycogen, which is a polysaccharide, and split it into glucose, and then to release those glucoses into the bloodstream. It doesn't release glycogen, and for that reason, this is wrong. Glycogen and starch are glucose polymers. Yes, polymer means many individual units, and the glucose are the individual units, so they are both polymers. Protein is a polymer as well, but of amino acids. Herbivores have larger canines. Nope. Taste buds are located on the papillae of the tongue. Yes, we know that. And CCK triggers the release of HCl. No, CCK is a hormone that's released by the small intestine due to the presence of large lipids, which causes bile to be released from the gallbladder. All right. Okay, I think I'm going to leave it at that point. That's quite a lot of review. I hope that helped you. I hope you find other resources that help you on Zerobio, my website, www.zerobio.com, or here on YouTube. And if you have any other questions, feel free to email me, see me in class, whatever. Good luck on your exams. See you soon.